So what is the purpose and hypothesis to our experiment?
The purpose of our experiment was to see how varying levels of nitrogen affect the growth of grass. We specifically chose Little Bluestem grass due to its native nature in the area. We hypothesized as a group that increased nitrogen levels will increase the height of the grass.
For this project, we decided that there would be 4 different environments, each with a different value of nitrogen. Each environment has 7.62 centimeters squared pots, each with 6.35 centimeters of topsoil. Each environment has 12 pots.
148 of the Little Bluestem grass seed was added to each pot. We made 4 different nutritional compounds that were composed of 1 gallon of water, 22.5 grams of baking soda, which is NaHCO3, 7.5 grams of Epsom salt, which is MgSO4, and the varying amounts of ammonium hydroxide, NH4OH. The first environment contained 0 milliliters of ammonium hydroxide, and the second environment consisted of 0.6 milliliters. The third was 1.2 milliliters, and the fourth 2.4 milliliters. 50 milliliters of the nutritional compound was added to each pot. The pots were placed in the McKinney greenhouse for 34 days and were watered every other day. Measurements of the plants were taken once every 4 days for the first 30 days. During the 34 days, pots were moved frequently in order to give similar sun exposure. Measurements were taken with a clear ruler from the base of the grass blade to the tip. After 27 days, we thinned the pots to 3 blades of grass in order to have the same sample size in each group. The stems were pulled and were counted, placed into bags in order to later calculate germination rate. Once thinned, measurements were taken every day for 4 days. After further growth, we then removed the grass from the soil and placed them in a drying oven for 24 hours to measure dry weight.
Our data showed a small correlation between Little Bluestem grass height and different levels of ammonium hydroxide. Though the initial growth rate from germination was very similar, around day 27 the nitrogen levels began to take an effect on the grass height. Germination rate showed highest under low nitrogen conditions at 11.3%, while lowest under normal conditions at 7.8%. Low nitrogen levels held an average blade height of 6.016, while the other conditions lagged slightly behind around 4.8 to 5.5 centimeters in blade height. When entering into the 4th week of growth, a more clear separation between environments began to appear, showing 0 and low nitrogen level pots having the tallest grass. Leaf mass was highest in the normal nitrogen environment at 0.008275 grams and lowest in the low nitrogen environment at 0.001469 grams. Our data for our project did not support our hypothesis. We thought at first that increasing nitrogen levels would increase germination rate, leaf mass, as well as increase the plant height, but our data showed otherwise. What our data did show is that Little Bluestem grass under low nitrogen conditions grew the highest as well as had the highest germination rate. A study conducted by Whedon and Tillman supported the trend that is shown in our data, and Little Bluestem grass actually was a better competitor under low nitrogen conditions in their study rather than high.
According to Salvar Dukas, a large increase in ammonium levels can lead to a depletion in other necessary nutrients for plant growth, such as potassium, calcium, and magnesium. A decrease in these nutrient levels leads to a decrease in photosynthetic rate. A study conducted by Anderson also supports this claim. Anderson tested Little Bluestem growth in fumigated and non-fumigated soil with manipulated levels of nitrogen, phosphorus, calcium, and magnesium. It was found that the nitrogen treatment and deionized water control had significantly lower levels of colonization compared to the phosphorus and base treatments. The reason Little Bluestem grass did not grow as expected may be due to the depletion of other necessary nutrients needed for optimal plant growth.
Our group may have been able to gain better results had we manipulated different nutrients such as phosphorus, which is critical for the growth of Little Bluestem. Looking back, we should have taken into consideration how much pH changes with the addition of ammonium hydroxide and sodium bicarbonate to try and keep the pH as close to 7 as possible, keeping in mind that nutrients are also bound by pH changes. Habitat restoration teams can take this data into consideration when looking at what types of grass to grow in native grasslands. Nutritive quality of Little Bluestem states that this grass is a pioneer species as it grows well in poorly nutrient soil and shallow, steep, or disturbed landscapes. It is also resilient in drought conditions, making it a top contender for restoration grasses.